So what I thought that I would do is give an overview of the Wellstone Centers, how we're organized and how we collaborate with groups not only here at UMass, but in other locations, Boston Children's Hospital, Ohio, Johns Hopkins, and then highlight some of the recent projects that we've been working on and accomplishments, and then tell you a little bit about the clinic here at UMass. So, oops. Oh, we don't need that. Sorry. Okay. Just trying to go back here. Just got a little bit ahead of myself. So the Wellstone Centers are translational research centers for muscular dystrophy to develop therapies. And these were originated through the MD Care Act by Congress and set up in honor to honor Paul Wellstone, the senator from Minnesota. He was both political persuasions really appreciated him. He died tragically in a plane crash in 2002. And in honor of him, these centers were named Wellstone Centers. There are, we're now entering the 11th year of these Wellstone Centers. So every 5 years, each center gets evaluated and in a competitive process. And this will be the third cycle for our Wellstone Center here. But I just wanted to point out that these centers are located across the country, but we all talk to each other, we collaborate, we share both reagents and ideas and plans for how to attack various different muscular dystrophies. Each center has its own area of expertise and emphasis. So for instance, our Wellstone Center is exclusively focused on FSHD and developing therapies as well as understanding basic mechanisms. There's a group in Seattle that focuses also on FSHD and Duchenne muscular dystrophy. The group at Rochester with Dr. Tawil focuses on myotonic dystrophy, but obviously he contributes tremendously to FSH muscular dystrophy. There's a group in Iowa that focuses on what are called sarcoglycanopathies, and then a group in Dallas that's focusing on editing of the genome to treat Duchenne muscular dystrophy. And in Florida, there's a group that studies fibrosis and how to counteract the destructive processes in muscle. So each group has something to bring to the table, and these groups get together once a year to share their ideas across different diseases and technologies. And so it's really been exciting. I'm a relative newcomer to the FSH field. I've been a neuromuscular neurologist for 25 years, but I've worked a lot on ALS in the past, which I still work on, but I got really excited a couple years ago with the group here when Charlie Emerson came, and we just saw this tremendous coming together of the field to focus on DUX4, and the fact that I was seeing patients in the clinic got me very excited, because we can learn a lot from whether it be a blood sample or a sample of muscle or a skin biopsy. So to me, that's very exciting to be involved in this. So the idea of our Wellstone Center is to focus on the molecular, the genetic, and epigenetic causes of this disease, and try to think about how can we counteract that? What kind of therapies can we envision? How can we cooperate with other centers and the biotech industry? And we're lucky to have Fulcrum represented here today as well. That is a company working on a therapy for FSH. The idea is to bring together the patients, the clinicians like me, the science investigators, and companies to try to accelerate our progress toward designing these therapies. And in addition, one other goal of our center is to provide training for young scientists. And you'll hear talks from both Alec and Caitlin after lunch. They're some of our trainees who are, have been instrumental to the program. So this is our Wellstone team. So I just discovered that in PowerPoint last night, so don't worry, that's the only slide I'm doing that on. Anyway, so this every year or so, our groups meet here at UMass and spend 2 days just intensively going through data and unpublished data, experiments, and brainstorming on what are we going to do next, and how does this result relate to the next experiment. And so this was from a year or two ago. This is Charlie Emerson, who's the director of our center here. This is Lou Kunkel, who is at Boston Children's. He's a molecular geneticist who discovered the Duchenne muscular dystrophy gene as well as the SMA gene, and he's been tremendous in various aspects of neuromuscular disease. Charlie is a distinguished scientist who is one of the world's experts on muscle development and the formation and function of muscle. And so his insights have been tremendous. Catherine Wagner, many of you probably have heard her or seen her at FSH meetings. She's at Johns Hopkins at the Kennedy Krieger Center, and she did the bulk of the clinical studies in the first 10 years of our Wellstone, and provided the muscle biopsies and many of the tissues for the studies that have been done so far, and is just outstanding neuromuscular clinician and scientist. And then there are many other members of the team here. Scott Harper is another principal investigator from Ohio, who I'll be mentioning about a little bit later. So the point is we get together, and it's not just our research teams, it's also representatives from the NIH who helped direct the program, as well as the FSH Society, and we're deeply indebted to Dan Perez and the FSH Society for helping to bring the patients together, and especially Dan for just advocating for funding for FSH and accelerating the science both at the national and the international level. So we get together, and this is just, I want to just spend a minute. I've already introduced a few of the people, but this is basically to give you an idea of that we're not sort of on an island here at UMass. But I wanted to give you a sense of the different range of expertise that each group brings to the table. So I mentioned Dr. Emerson and myogenesis. He also was instrumental in setting up what we call these xenograft models. So you can take cells from a muscle biopsy from a patient, and under the right circumstances, you can transplant that into a model organism that you can then study how the muscle behaves in a living animal as opposed to in a tissue culture dish. The biology is very different, and it's very important to study in an animal. And so he and Catherine Wagner helped develop this method where they could grow cells obtained from a muscle biopsy, and then transplant them into mice that would then take up the muscle cells and actually grow small little human muscle in the leg of a mouse. And with that type of an experimental setup, then we can do all sorts of manipulations to understand what's going on. How is the FSH muscle different from normal muscle? How does it respond to injury? All these sorts of possibilities. Oliver King is here today. He has a mathematician's background and is an expert in genome biology and bioinformatics, and has been instrumental in helping to understand what are genetic differences among different FSH patients and unaffected controls that might explain why some people get more severe disease and some people have less severe disease. And why it may affect one muscle and not another muscle. So having just a blood sample that gives us DNA gives us a tremendous amount of information for him and others in the group to analyze, to try to tease out, are there certain genes that might be protective that some people have? Because if we knew what those were and we could develop a drug that boosts the activity of that pathway, then that could be really beneficial. And then in this recent, just this year, 2 more members of our faculty here at UMass, Anastasia and Jonathan, have joined the group. And they are experts at what we call oligonucleotide-based therapeutics. What that is, and they're on the 4th floor of this building in what we call the RNA Therapeutics Institute. So it's an amazing, it's probably one of the most powerful groups working on RNA in the world, and they happen to be right here. Craig Mello was a Nobel Prize winner who discovered a way to silence RNAs and got the Nobel Prize 12 years ago for that, and is here. And since then, the whole department in that group has grown, and we've been in touch with them. And basically, these 2 investigators, they're expert at designing strings of nucleotides that can be injected that home in on an RNA of interest. So in our case, we're interested in the DUX4 RNA. We want to get rid of it. These nucleotides can home in on it and then neutralize the RNA before it can get translated into protein and cause damage. And so we've been starting to collaborate with them on what their expertise is designing the right chemical moieties so that the nucleotides don't get degraded too quickly. They'll last for weeks or longer in the body without being destroyed, and also that to help deliver them to the muscle more effectively than if you just, so there are certain chemical modifications you have to make to get this to happen, but they're expert at that. And so that's something we're actually testing, and you'll hear some more about that. This is on auto advance, I'm sorry. Okay, so, and again, I'm in the FSH clinic. I'm interested in helping to understand what things can we measure to see how well a patient is doing or might be responding to a therapy. And that's clearly going to be important as we get agents that we might be able to try in patients. We need to have some measure to know, is it making a difference? And so there are many people working on this. Dr. Tawil at Rochester and Jeff Statland are spearheading how to set up clinical trials, and Dr. Wagner as well. And so what I want to do is make sure that our clinic sort of corresponds to the guidelines that they've set out, so we can all pool all the data together and really learn something from our patients. And I also have a basic interest in what's known as single cell biology. So there are actually, and I'll mention that later, there are ways of taking individual cells from a muscle biopsy and looking at how genes are perturbed and how the expression goes in individual cells, which gives a lot more information than if you take the whole tissue and just look biochemically at it as a whole. So that's our group. Some of you may have heard from Dr. Kunkel and Dr. Leck. Is there a way to turn off this auto advance? I'm sorry. Their expertise is, Dr. Kunkel has also set up zebrafish modeling. So zebrafish is a model organism that has the advantage that the embryos are transparent, and for the first week or two, it's a transparent little fish that they can visualize gene expression, and they can actually put hundreds of fish in different little wells in a plate in culture, and apply a different drug to each well and test what effect that has on the muscle in a little larval fish. And so they've developed a fish that will express the DUX4 and has pathology. So that offers a really good platform for testing a larger number of drugs more effectively. Angela Leck, who was a postdoc in Lou's lab, who's now at Yale, she did a really interesting genetic pathway screening analysis that I'll talk about in a little while. I mentioned Dr. Wagner, who is a an expert in the clinic and has also been doing more with imaging and MRI to look at, to actually visualize the muscle structure in patients. Two newer investigators are Dr. Johnson and Dr. Weiss at the University of Utah. These individuals have a background in population-based genetics and disease modifiers. And so what they're doing is they have access to a very large family in Utah with FSHD1 that from my reading, there are over 1,000 members of this family, probably even more by now, that all descended from a single individual who emigrated from England and settled in the Salt Lake City area in the mid-1800s. And so what's interesting about this family is the number of repeats, that influences the severity of FSH has stayed fairly constant among all the family members. So that's sort of a constant, and yet there's a lot of genetic diversity among the individuals and a lot of diversity in the phenotype or how severely affected some people are versus another. So it gives us a great opportunity to look again for other genes that might be modifying the effect of that single FSH disease insult. So they've got the tools to analyze that and again are focusing on the modifiers. I mentioned about Dr. Harper. His expertise is delivering genes as therapies. So if we knew of some gene that if we could just turn it on or turn it off, then that could be a potential therapy. And so there's many people working on trying to just downregulate the DUX4, but potentially could upregulate other proteins that may be helpful. And he's produced an inducible mouse model, so a new mouse model that expresses DUX4 at just the right level that you start to see pathology over months rather than too much DUX4 all at once. And his group has also looked at the DUX4 protein itself, because we don't know a lot about what, you make the protein from the RNA, but what turns on the protein, or what does the protein interact with? What makes it affect its function? And are there other ways we can just block that even if the protein is there? I mentioned the Seattle and Rochester Wellstones. We have some overlap, and we've invited, and we talk regularly with Dr. Tapscott and Dr. Tawil, they've come to visit our campus for in-depth discussions, and so it's really exciting to be able to work with across the different centers. So I just wanted to spend a little time to give you an overview of the different groups. But clearly, you guys are the backbone of our Wellstone Center, and the ability to learn from you as patients and also, perform the research and hopefully very soon in the future be participating in clinical trials is utmost. And then in gatherings like this where, we can get inspired by meeting you, and the trainees in our group can also be inspired, there's no substitute for that. So this is the way our center is organized. There's our center directors that I mentioned, and they are in close communication with an advisory committee from NIH. And we have administrative resources so that we can set up web meetings. We meet once a month, the different groups at locations. We have a web meeting where 2 or more people share their latest data, and everyone's sort of up to date on what's going on. And then yearly we have a bigger meeting where everyone gets together. But the core of our organization is to bring together the investigators and the patients through the help of FSH Society and other groups that can keep us in communication. And our goal is to be able to help the patients and get going on the clinical trials, but also to generate some of these resources that can be useful for research, not only in our group, but around the investigators around the world. And as I mentioned, the idea of training some of the younger scientists is really important as well. So what are some of these resources? So clearly, what's important is FSH is such a variable disease. There's so much diversity among different patients. The more patients we can get materials from, the more we can learn. And so at present, in the past few years, almost 200 individuals have been enrolled in the studies to collect either blood for DNA, which can then be used to do gene sequencing and really find some of these modifier genes, or in some cases, muscle biopsies. And we focused on biopsy of the deltoid and the biceps. The deltoid is less affected in FSH. The biceps is more affected. And there are a number of individuals where we are able to get biopsies from both of these muscles, so that we could compare, what's different about one muscle versus another, and learn, why is one muscle more susceptible than another. And there are different groups here. We have family cohorts where there's an affected individual with moderate weakness, and then an unaffected first-degree relative. Controls are very important, so everyone can participate. What's really interesting is there are some families where both two people may have the same FSH deletion, but one has a more severe and one has a less severe, and we call that clinically divergent. And those are particularly potentially very informative. So we're always, we're interested in obtaining whatever materials we can. From a muscle biopsy, we can look at it under a microscope and look for pathology. We can study the biochemistry and look at what proteins are altered, and we can look at the RNA. We can also take muscle fibers, and Charlie's group has done work with physiology, how well they contract or they respond to injury, things like that. And then one of the real cores of this initiative is to collect from any muscle biopsy. We can take some of the cells, put them in a petri dish, let them grow, expand them to enough where we can then put them in individual vials and freeze them down so that we can send them to other researchers around the world who have a question they want to ask using just the cells. And we can also, from either muscle or from skin biopsies, we can generate stem cells that can then be induced to proliferate and then form muscle, again, for future research studies. And potentially for treatment in the future. So I'm not going to go into too much more detail about the specific projects, but basically these resources feed into some of our main projects. I already mentioned several times about looking for genetic modifiers. So again, all these groups that we're working together to try to think about how can we identify these. And the addition of the Utah family, I think, will have a big impact on that study. The drug discovery arm is interested in identifying pathways that are important, like the CRISPR screen, and seeing if there are drugs that can be repurposed to affect a difference in one of the cell models or animal models. And in Project 3, preclinical development, this involves using those animal models, zebrafish or mouse models, to test ideas that come out of the other projects. So there are numerous sub-projects here, but they're all sort of integrated together. And out of these come the cell and animal models that we can share with other people. And what's really exciting is that we have other Wellstone collaborators in the network, but also industry has gotten involved as well. So we're working with Genea Biocells. They have a great protocol for turning these stem cells down the muscle pathway, and so we can generate a lot of cells using that protocol and do studies from there. So just to give you a few of the highlights of recent findings, this was a paper that just came out from Lou Kunkel's group, and this is their zebrafish model. And to me, what was interesting was, so they're able to turn on DUX4 in a developing zebrafish really early on. And what they found was that the DUX4 was on for a period of time, and then it slowly went down as the fish became an adult. And yet it was later on that they started to see pathology, even when DUX4 was off. So basically what you're seeing here is that early on, after 2 weeks, there is some pathology here. There's loss of what you see here. This is normal muscle over here, and this is muscle that's being replaced by fat or collagen tissue. And if you look later out, this is after the fish is fully developed, and we look in a cross-section through the fish muscle here, what you see in a control is just this normal muscle. Here, they saw accumulation of collagen or fibrosis that you might see in a patient after muscle's been damaged. And they're seeing it, it's not just happening everywhere, it's asymmetrical and it's focal. So it kind of resembles what we see in the patients, where it's not like it happens everywhere or symmetrically. And so it's very interesting that this was happening at a time when there wasn't really much DUX4 expression either. So it raises the hypothesis, it still needs to be tested further, that there could be some early consequences of DUX4 expression at a very developmental stage that can manifest later on in the adult. And I mentioned briefly about the CRISPR screen. This was, Dr. Leck did this experiment, and basically it's a novel method to, you take a bunch of cells, and with this CRISPR reagent, you can randomly knock out an individual gene in individual cells. So you basically have many cells where a different gene is knocked out in each cell. And you don't have control over which cell gets knocked out, but it doesn't matter, because then you turn on DUX4 and you just monitor this, you just let the cells grow, and the ones that are resistant to DUX4 toxicity will grow out in the culture. And then, then you can, once you have those cells that have proven themselves to be resistant to DUX4, you can go in and sequence and find out which gene was knocked out in that individual cell. And you get a list of genes that if you knock them out, they improve the disease, potentially. And so that's what Angela was able to do. And so she identified some specific pathways and some known drugs that affect those pathways. So now they're testing those, they've been testing those in the zebrafish model. And so it's a promising approach that might provide some relevant drugs to try in other FSH models and perhaps in clinical trials. This was Scott Harper, who just last month presented their new mouse model, which they were working on this almost 10 years because DUX4 is so toxic. They had to titrate the levels just to the right degree, but they made a design where just giving a chemical tamoxifen at just the right dose, they can induce the disease in the adult mouse. And so now we have at least 3 FSH mouse models, because there's Dr. Kaiba's model, Dr. Jones's model, and now Dr. Harper's model. They're all slightly different, but you know, it's great to have more than one model that can be used to test these different approaches. And so in Scott's mouse, he showed that so here's an example of a normal muscle here, and here's a muscle of one of his mice where they didn't give the activating drug. And here, these are 2 different muscles from a mouse where they gave the drug over 3 weeks, and they were starting to see damaged fibers in these particular muscles that they looked at. And they actually measured the strength of these muscles, and they were weaker. So they've got a protocol now where they can induce this, and then they could on top of this, they could give a treatment and see if it's effective. So after we finish lunch, we're going to hear 2 talks, one by Alec in the Emerson lab, who's here in front, and another by Caitlin. And so you'll hear more about some of the topics I've introduced. Dongsheng is another postdoc in the lab he's been working on turning the stem cells down that human muscle myogenesis pathway to expand them and make muscle. And it's really interesting work that basically we're using this protocol from Genea BioCells, where basically you start with a stem cell, and over about a 10-day period, under the right growth conditions, you can trigger the development down a pathway toward the development of muscle. At this point, you change the growth conditions and actually start to commit and differentiate these cells so that they're small single muscle cells. They haven't fused yet or fully differentiated, but they start lining up with each other. And then in another week under in differentiation media, they actually can fuse together, form these long muscle cells in a culture dish, and actually start contracting. So it's a way of sort of going from a single set of stem cells that we can, again, we can get that from a patient from either a skin biopsy or a muscle biopsy to a whole dish of active muscle that we can either study or perhaps in the future, you know, think about ways of transplanting back into a patient. This is what they look like. These are the stem cells here. This is the first set, the first stage here. There's the second stage, and then once they've actually fused into muscle. I'm going to pause right here and just see, it's a lot of information, but are there any questions so far?
My husband is from I'm curious to find out if he's part of that family.
Oh, he may be. I mean, Kevin Flanagan did, well, the family was first reported like in the 1950s, and there was a big, there was a big comprehensive paper that described many family members in detail. And then also Kevin Flanagan about 10 years ago summarized an update, and you know, that's where there's like over 1,000 family members, but okay, yeah. Well, they may well have, again, our collaborators there would probably have access to the records. So if he wants to give them a call or the.
And another thing, my son has the relation imaging and has 37 representative symptoms, and my husband has the relation in his stomach.
And when did your, so the question is, I'm just repeating for the people who are webcasting, that you have a son who's 37 who has the same genetics.
Same genetics, he was diagnosed in 2000.
Does he have any symptoms? None at all. And your husband, what age did he develop symptoms?
15, diagnosed at 16.
Okay. Yeah, so clearly, you know, to look at even just a DNA sample from those two, would be really informative, because that's the idea, is we want to try to understand why, you know, why is your son, you know, protected? It could have been that he got a gene from you that is protective. So, yeah, yes. Yeah, and there's another question. Yeah.
You mentioned that there was some muscle atrophy slides, some zebrafish or some animal. You said that happened.
In that zebrafish, the DUX4 is on fairly early. So some of if there's early changes, it could be because of DUX4. But what happens is later on, the DUX4 protein becomes undetectable, and that's when they start to see some of these other changes. So it raises a question whether something about, you know, there's some memory where DUX4 has some consequence early on that doesn't really show up till later on. Yeah, the DUX4 protein. Yeah, question?
On the mouse model study, you mentioned tamoxifen as an agent of interest.
Yeah.
Aromatase inhibitor used for cancer.
It is. It's used in breast cancer treatment. There's nothing necessarily special about that chemical, but the way it's a system that's been in use for a number of years now, where there's a receptor for that can then turn on a gene that will then activate whatever other gene you engineer into the right place in the mouse. So it's been used a lot in the research field as an activator, because it's not normally, it's less likely to be just turned on randomly unless you give the chemical, right? Yes.
Does
this question about the zebrafish. This DAX4, is it the soluble DAX4 that's detected, or is it just like is it slowly self-assembling?
Oh yeah, that's an interesting question. So the question is, in these models, in the zebrafish model where you're expressing DUX4, does it stay soluble, or does it aggregate into complexes? And it's been difficult to study DUX4 because there haven't been great antibodies. The studies I'm aware of show that when you have DUX4 mRNA, that gives instructions to make the DUX4 protein. It's expressed at very low levels, and so it's made outside of the nucleus, but it acts inside the nucleus on the DNA, so it then can be transported into the nucleus. And so we often, when we stain, we see it in a nucleus. But if it's made in the cytoplasm, muscle cells have many nuclei. They're big cells, and they have nuclei sort of lined up together. So you can actually see a gradient where maybe it's made in the cytoplasm in one location, and there's a lot of it in this one nucleus, but the next door neighbor nucleus may have a significant amount as well, but less, and then less in the next nucleus. So it's almost like it could diffuse and reach another nucleus as well. It's such a low level, it's hard to see if it's really in an aggregate form. It's thought to, what it does do is it binds to DNA. It's got these elements in it that home in on a specific sequence in DNA. And so and I don't know if off the top of my head, but it's like ATTGGCCT, something like that. And turns out that sequence is in front of hundreds of other genes in the genome. And so when DUX4 goes into the nucleus and binds there, it facilitates turning on all these other genes. And so it's not hard to imagine why that could be maybe not a good thing in a cell. So it's a very powerful, what we call transcriptional activator. And yet it's not a malicious protein, even though we don't want it in our muscle. It's actually, none of us would be here if it weren't for DUX4, because it acts normally in the very early stages of development. It's involved in what's called zygotic gene activation. So when you've got a fertilized egg, and it's just like at the 1, 2, 4 cell stage, just starting to form an embryo, DUX4 and some of its targets are really key in turning on all the hundreds of genes that get the embryo going. So it's kind of like an ignition switch for the embryo, but it gets turned off very rapidly after that. That's its only job. It gets turned off, and then the rest of the embryo develops. And it normally gets turned off by being packaged in this chromatin that normally then keeps it off for the rest of your life and keeps it off in your muscle. But of course, with the deletions of the repeats, there's a tendency for that to open up again inappropriately. So what's bad about DUX4 is its dysregulation, but not the protein itself. And so that's what, there's a lot of just understanding about what activates it and things like that at the protein level, I think is very interesting that we need to learn more about. Yeah. Other questions?
Is there any
Yeah. Yeah, that's a really interesting, that's a very interesting point. And I believe Anna Pakula in Lou Kunkel's group has gotten some support just recently to look at the estrogen question. Because, from my perspective, there's so much we can learn from the clinical observations like that. I think, so there's bound to be more coming out soon from that. Well, we just have to understand what's going on, and Yeah.
You don't want to
Okay.
recommend self-medicating.
Not yet, not yet. Yeah. Okay. Well, so I'll just go on in the interest of time. And so I just wanted to briefly highlight, so what I'm really excited about is there's so many different advances on different fronts that are happening at the same time. So to me, that's really exciting. So I mentioned Dr. Statlin and Dr. Tawil. So they're working on getting us ready for clinical trials. You know, what are we going to measure, and what are we going to record when I see a patient in the clinic? What kind of useful information are we going to gather over time so that we can define the natural history in as many patients as possible of this disorder and get a sense of, you know, what's a significant improvement or not. So that's going on one front. There are a number of therapies that are designed toward just in general improving muscle function. And so these aren't necessarily targeting FSH in particular, but for instance, there's a human growth hormone and testosterone, if you want to try that one. There's a clinical trial at Rochester testing these ideas. There's another interesting one that Acceleron is working on using a protein that inhibits a skeletal muscle inhibitor. So it's kind of a double negative, but this modified follistatin, it helps to block the function of myostatin, which keeps muscle from growing bigger. And so what they're doing, they have this protein. It's a protein, so they have to sort of inject it in muscles. So they're injecting it in a leg muscle and an arm muscle. And so far what they've seen is that it's safe and it increases the volume of muscle. So that's potentially very interesting, although it would be a little cumbersome in that every muscle has to be sort of injected to see the benefit. But if that is beneficial, there could be workarounds for that, because there are other people working on if we have a protein we want to deliver, how can we deliver it like with a gene therapy sort of approach? So it's going to be informative regardless. And then there's a big category of strategies that are targeting DUX4 itself, where we know this is really specific to FSH. And you know, I don't have time to go into all of these, but there are a whole group, there are companies and individual investigators working on, you know, how can we lock down the gene and prevent it from being opened up and accessed and activated. And like Fulcrum, I think, and others have reported compounds that help block the turn on of the gene and can modulate these repressive pathways. The DNA then makes an RNA that encodes DUX4, and some of our projects here are focused on counteracting this RNA. I mentioned earlier the antisense oligonucleotides. They home in on this sequence and neutralize it. And so if we can just deliver enough that lasts long enough, that could have a big effect if there happens to be some transcription and DUX4 mRNA made. And then the next level is RNA gets converted into proteins. That's where the DUX4 protein itself is made. The RNA are the instructions for the ribosome to make the protein. And there may be, Scott Harper's group has been looking again at how the protein gets activated and how can we maybe decrease the protein levels and prevent it from accumulating or triggering its downstream effects. And then there are many known targets, and there may be specialized treatments that maybe there's only 2 or 3 of these targets that are the most important to treat, and we could design a drug for each of those and have a measurable impact on the disease. So there are many different levels, and it's great that, you know, different groups are working at different levels, because there's nothing to say that you couldn't combine these treatments, because they're all acting on different aspects. And so it's a very exciting time, in my opinion. So that's why we have our FSH clinic here. It's twice a month on Thursdays. So my colleague Kate and I see patients. And again, we want to try to include as many patients as possible so that it gets to be part of the national database of information.
So what do you do with those patients?
Well, there are clinic visits where we find out about the history. We want to find out about any other family members. Pardon me?
Just like the intake when you first treat them.
Yes, yeah. But we also have, it depends on, each patient may have special needs. So we have collaborators. Well, let me just point out, yeah. So we have, we want to expand this to include as many of you across New England as possible. We have this biomarker study, which you can read about on the web. But basically, if we have a blood sample, we can do genetic testing if that's needed. And then we have a rehab, if people are local, we have a rehab hospital that's nearby where there are physical therapists who know about FSH and may be able to give some advice. You know, it's a problem in that not that many physical therapists, you know, there's a real need for more specialized knowledge. And so that's what I'm working on, trying to get more expertise in that area, because it would be nice to be able to offer that directly. And we're really lucky because there's a pediatric neurologist, neuromuscular specialist, Brenda Wong, who came to UMass from Ohio in the spring, and she has one of the largest Duchenne muscular dystrophy clinics in the nation. She sees the Duchenne boys, and they fly in from all over the country to see her. And so she's got a physical therapist who's just excellent, who knows a lot about, you know, the different types of muscular dystrophies. And we want to try to see if we can get maybe a second person and share resources and, you know, have it right here on our campus. But Fairlawn is a really good rehab hospital right nearby as well, that they're happy to see patients with FSH. There also are often questions about genetic counseling, and then we can do vision screening or pulmonary function, whatever people need. But I think one of the things I'm excited about is we want to try to participate in this network that will be probably the first to be involved in clinical trials when they become apparent, that Dr. Statland at Kansas has set up, initially through funding from FSH Society and the MDA and now the NIH as well. And he has about 7 centers now, but the plan is to have at least another 10 centers in the next year or two. So since there's not one right in the Boston area, that would be really nice to be able to be included in that network. And so, so that's another goal here. This is just, you can download this, but this is just some information about this study. So we're obviously interested in patients with FSH or their family members, whether you have it or not, or if you're a control, we need control samples as well. And there's more information about it here, but the goal is to have the DNA to look at biomarkers, but also to potentially have skin or muscle tissue that we can use for these other studies that I already talked about. And then my particular interest is that one of the things we can do now in the clinic is muscle ultrasound. So this is just a little laptop that we have in our neurodiagnostic center where they do electrical studies of the nerve and muscle. And nowadays, the latest machines have these little ultrasound probes. So you can actually just place a probe on top of the muscle. It's painless, non-invasive, and you can move it around. You can sort of see in real time, you can see the structure of the muscle under the skin. It's called the echogenicity. So it's sending ultrasound waves, and things are bouncing off depending on the structure. And if it's been replaced by fat or fibrosis or things like that, it's readily apparent. And without having to do any surgery or anything like that, you can see you can document change over time. So this might be a really good something that one could follow that would be more objective than just measuring strength in a patient who's in a trial. But we need to define, you know, what's the range among our patients. So these are just some of the advantages. The only disadvantage compared to something like MRI is it doesn't look quite as deep in the tissue. So you're just looking in the muscles that are closer to the surface. But I was over in the lab the other day. So this is actually a cross section of the thigh muscle of one of my colleagues, just we're moving the probe. We're looking in cross section. And it's hard to, it's basically where you see light areas, that can be sort of like fascial separation between individual muscles. And the top would be where the skin is. So this would be the skin right here, and then there's fat, which is dark here, and then this would be the muscle tissue. But I'll just play it just so you get a sense of how it's just real-time, live, dynamic. You can move it around, and you can basically get a view of the muscle. And if we saw an unusual area, then we could say, oh, that might be a good area to get a muscle biopsy, because we might learn something. There might be some inflammation or fibrosis or something that would be informative. So it's potentially allows us to get muscle in a more informed way rather than just randomly choosing a biopsy, which in FSH, it can be so patchy, you want to make the best use of your one shot at a biopsy. So, and I'm almost done. And this is just a comparison of, like, here's an MRI, beautiful pictures here. It just takes longer to arrange this, and so, but here's an example. This is a thigh muscle. Here's the femur, the bone in the thigh, okay? This is the front. This is the muscle right on the top of your thigh, the rectus femoris. This is normal looking. What's that? Yeah, it's kind of the same idea, yeah. And then this is the same view. So for the ultrasound, you're really kind of just looking at this area here. So this is the same muscle here, the skin up here, the fat, and this looks pretty normal here. Here's an affected muscle in FSH where, again, in the MRI, you really can see, like, these are normal muscles, and then you get this one muscle that's totally dystrophic. It's a different signal. All the white on the outside here, that's your subcutaneous fat. And, you know, and that's variable. This person has a lot less fat outside compared to this one, but that's normal. I mean, it's variable, but what we're really interested in is in the muscle areas here. This muscle has been totally damaged here. It's this bright signal on the MRI. It should be dark. And then we can also see an abnormal structure here in the ultrasound. It's too bright. And when you look closer, you can see sort of more fine structural differences. But ultrasound can also offer some advantages, because there's sometimes when you look on the MRI, like this muscle looks pretty normal on MRI, but here you actually, and I can't point it out to you, but you actually can start to see structural abnormalities consistent with fibrosis, that the MRI is not very good at picking up fibrosis. So the main point here is these are complementary, and you know, the ultrasound might be even a little easier to use in the clinic, because we could do it at every clinic visit and just sort of monitor. So last slide or two here is just this idea of single cell biology. And I'm really fascinated by this. The scientist in me is really fascinated by this. But this is basically what we can do is we can take single cells, say we get a muscle biopsy, we can dissociate it into single cells, and we can do this also from cells that we've grown in a culture dish. So we can imagine just a suspension of single muscle cells, and they're all unique. And we want to really statistically examine them in detail and determine, you know, there may be only 1 in 1,000 that expresses DUX4, but if we find that one cell, that's going to have a lot of information in it. And if we just grind up the whole collection of cells and try to do biochemistry, it's going to be lost. We'll never see it. But with this technique, what you do is you have this suspension of cells, and if you see these little squiggles, it's a microfluidic device where, see these little squiggles here? These are channels where from both the top and the bottom, single cells are flowing through, okay? This is happening very fast. Single cells are flowing into this device. In addition, there's a channel right here where these barcoded beads are flowing in as well, okay? And so as this is all happening, there's also oil coming in here and here that, if you can imagine, it's pinching off these little droplets. So you see, it's all flowing toward the right, but as the oil comes in, it pinches off a new droplet. So we get these tiny little droplets that are then just kind of flowing along. And what we aim for is to get a single cell and a single bead in a droplet. And when that happens, a chemical reaction occurs and the cell breaks open, and all the RNA messages from that cell get bound by the bead, and then they're all collected later. And they're analyzed in mass, in bulk, but each bead has its own barcode on it. So later on, we can get the data back, millions of reads of the RNAs to identify which RNAs are there, and we can assign them back to individual cells. And that gives us a picture. This is just sort of diagrammatically what happens is each little droplet is its own little reaction vessel. And the cell breaks open, the RNAs bind, and then we can analyze this very easily. We get a huge matrix of like 1,000 columns. Say we had 1,000 cells, like 1,000 beads that were unique, and then 20,000 rows, one for each gene in the human genome, and we get a count number for each gene in each cell. And from that, we can say, well, how can we group all these cells by which ones are most similar to each other? So this is the kind of plot we get. Each little blue dot is a different cell, and each gray dot, which they're all kind of running together, are also single cells. So this is maybe like 3,000 cells, and they're grouped on this graph so that any cell that's nearby has an overall similar gene expression pattern, okay? And so what we see here, this is from some cells that were in culture, is that we see there's two main groups. There's this small group on top and this larger group. And the bottom group here, these are expressing a lot of the factors we would expect in muscle-like cells, or cells that could form muscle, because these are the genes that we normally look for. These are muscle stem cell genes, and these are muscle differentiation genes here. And yet they're not all the same. There's quite a bit of variation. But then once we have this data, we can ask, well, I'm interested in gene X, and you can look at what's the expression of any other gene, ones that we wouldn't even necessarily think are related to FSH. Or we can ask, well, okay, I see this group here, but I don't see any genes on. I don't see none of these genes that I've looked at so far are on. And I can ask, well, are there genes that are on in this group but not in this group? And it can pull out the names for us. So then we can verify that, and we can verify that these are non-muscle cells that probably came from other cells that were in the biopsy. And are there other genes that are mainly on in this group here that we didn't know about before? So here's one that seems like a pretty good marker for this group, because it's on at high level in most of these cells. And so we can learn a lot about, how the cells differ from each other, and then what are some new genes we might want to be looking at. So that's where I'm going to end it here. And I really want to thank all of our sponsors, and especially everyone here for coming, and everyone else for listening. I think there's more food here, and I'm happy to take any further questions too.
I had a question, just for clarification about the FSHD clinic.
Yeah.
So, in addition to, you provide clinical care, right?
Right.
And then it's also an opportunity for people coming to clinic to participate in some of the research studies.
Right.
And so, it's a clinic where people can be coming for annual visits. Is that?
Ideally, yes. So the question is in addition to providing care and providing information about these ways that patients can participate, are we interested in following patients annually? And that would certainly be the case, because we want to, especially if we're coordinated with the other centers and we're collecting the kind of data that they would want, as I mentioned before, to pool together to define, what, can we define the range and the natural history of the condition for different people. then a yearly visit, or even, you know, even more often if they're local in the area, would be really helpful. I know Dr. Tawil has just finished a study where he would have several patients would come back on a yearly basis, and they would actually repeat the MRI of the leg muscle, and they'd look at the same area by MRI. You know, and that takes a lot of money because MRIs are expensive, but potentially we could do something similar, but just using the ultrasound. It would be very easy and inexpensive, and you know, hopefully provide some useful data. Yes.
You said you only have one shot if a patient's getting a muscle biopsy. Do you mean that you can only take one biopsy from one patient at any given time? Is that specific to what muscle you're biopsying?
Yeah, you don't really have one shot. It's just many patients. I wouldn't want to put patients through an invasive procedure if there's a low chance that it's going to provide information. But certainly, yeah, certainly one can do more than. And when Dr. Tawil did his study, what he would do is they would know from the MRI where they did the muscle biopsy a year earlier. And you do want to know if you've done one at a certain site. So you want to move away enough that you're not going to be picking up damage from the previous biopsy. But certainly, yeah, there's not really a limit to how many biopsies you can do. And you can do, there are different types of biopsies too. There are biopsies where a surgeon will open up the skin and get sort of a piece the size of like a long pencil eraser, and then sew up the skin and the fascia underneath. And that's what most people get for clinical diagnosis, because it's a big enough piece that one can make slides and really look at the pathology. But for the cell studies that I'm doing, it might not necessarily need that much tissue. And there are other approaches that Dr. Tawil has used where you go in with a needle and you can actually get enough tissue that way, and it's just a very quick procedure. So, but yeah, there's not any limit to one biopsy. Yes.
So, is there any hint as to what affects the rate of change? Seems like it's in steps. I don't know, like what?
Yeah, that's.
One person is this way, another person.
Yeah, it's a really interesting question. So the question is, it seems like from a patient's perspective sometimes that everything's going fine, or you're used to a certain level of disability, and then all of a sudden there's a jump down. And, you know, is that because something suddenly happened, or is it just that it's gradually worsened and that at one point you notice you can't do something like you used to? But I think from what a lot of patients say is they can notice a pretty dramatic decline over a short period of time. And there are older studies that have shown where they've actually done repeated biopsies where, like on the first few biopsies in a certain muscle, there was very little pathology. And then after the patient had a big decline, they did another biopsy and they saw all sorts of inflammation and other changes. And so we really don't know, but that is it's a very important question. We do see, a point I make in talks is that when you do a biopsy, even though the pathology may be very mild, you often see these inflammatory cells right around the blood vessels, and they're just sitting there. They don't seem to be damaging anything, but something has attracted them there. And so that's a clue in my mind. Something about FSH has attracted these cells to that location. Even though they're not doing anything yet, it's possible they could be there for a good purpose. They're maybe doing some repair of things that we can't see on the slide, or maybe they're there, and if another hit comes along, some injury, they could be activated aberrantly and cause damage. There are some theories about, certainly in FSH muscle, people have seen increase in what we call oxidative stress markers. And so it might be that certain environmental insults could trigger a stress that under the right circumstances, you know, leads to additional damage. But I've seen patients where, you know, and things like exercise induce oxidative stress and mild damage. But from the patients I've talked to, you know, as long as you can exercise, that's a good thing in general, as long as you don't overdo it, you know, because muscles, if they're not exercised, they will get smaller. And it seems like at least one of my patients is a very avid with exercise, and it hasn't really, you know, as long as he knows his limits, I think it's helped him more than it might have harmed him. So but there's still a lot we don't understand. Okay, all right, well, thank you very much.